Dr. Thaxton, how did you first get interested in the origin of life?
When I was a graduate student in chemistry at Iowa State University, my subject matter was not origin of life studies. It was X-ray crystallography, and we were doing small molecules, not huge proteins or something like that. And there was a postdoc who was a postdoctoral student in our lab who was from Harvard and was in contact with some of the Harvard people doing origin of life studies. Well, it had been a fascinating idea. I had heard a couple of seminars on the subject, but never really particularly focused on it. And he mentioned a book that was a new book by a man named Dean Kenyon that was called Biochemical Predestination. And it happened to be in our bookstore, so I picked the book up, began to read it. I was very fascinated by it because I felt like that on first reading, it all sounded so solid, so, it made perfect sense. But as I began to reflect on it later, I realized that there were a lot of things that I had already learned as a chemist that it just didn't fit. And I had sort of puzzled questions, but no real approach to it. Well, I went to Europe for a year, and during that year I had a chance to reflect and think a lot more about the book. That was just after I'd finished my PhD in chemistry. And I had a chance to get some thoughts together more specifically on that subject. At the end of which time I wrote a little essay giving my response to basically Kenyon's thesis in the book and sent it to him because it was just a subject, interest, fascinating, growing subject. And his response was that, well, I like your criticisms very much and encouraged me to continue on with it. And that's basically where our book came from, was out of that original a response to Kenyon's book, The Biochemical Predestination. So it wasn't any lifelong search for how life began. It basically came during the end of my PhD studies and when the subject first entered my consciousness as to, oh, this is a subject that's in the traditional evolutionary mode that fits my background and training. I think I will look at it and see what I come up with. And that's basically how it happened.
How did you come to write The Mystery of Life's Origin with Walter Bradley and Roger Olson?
Well, I came to, after my time at both Harvard and Brandeis, I had written more on it just privately and developing my own thoughts. And when I came to Texas after Harvard experience, met Walter Bradley, who was also interested in the subject. As we discussed, found out we had very similar interests, but we seemed to be missing each other. He was an engineer, thermodynamics specialist, and my background was, I knew about thermodynamics, but had learned it as a chemist and not an engineer. And it just seemed like that in our communication, we were missing each other. And we discussed and eventually figured out that there was a slight adjustment and we could get our vocabularies where we communicate. By that point, I had already met Roger Olson, who we also collaborated with, and Roger was a student of Bradley's at Colorado School of Mines. And so the three of us began to discuss and find out that our materials meshed, and so the three of us joined up to write that book as a critique of chemical evolution.
Was there an advantage to having the three of you collaborate on the project?
Oh, absolutely, because the field lends itself to an interdisciplinary study. As you might imagine, if you're going to discuss the question of where life came from, that requires knowledge a bit about chemistry, geology, physics, all the different facets of what's going on with other planets, the atmosphere. Wouldn't hurt to know some biochemistry either. And just a variety of different disciplines. So astronomy or astrophysics, all these different disciplines are coming in together. Well, Roger was a geochemist, So he covered the geology, geochemistry, the conditions of the early Earth in our analysis. I did the analysis of the experiments, the chemical experiments and simulation processes, and the chemical part of it. And then Bradley did the discussion about thermodynamics. And then the three of us discussed about the rest and the philosophical discussion part of it.
What is the primary argument of the book?
The argument of the book that we put together was to look at the theory itself from the assumptions that are made by those who present it. We wanted to give a legitimate critique of this field, not stand aside and throw stones at it, as you can do if you're any kind of a critic. You can always throw stones at something. This was not our objective at any point along the way, either from the beginning or anywhere along the way since. The idea was to get inside the minds of those who do the research and to interact with their minds as well as interact with them as individual people to find out what they've done, how they did it, read the fine print in the research papers, and try to see what were their assumptions, how did they do it, and then from within arrive at a legitimate critique. And that's what we tried to do.
Why do you consider the origin of life to be a mystery?
I believe it's still a mystery because the approach has been too crude for the complexity of what we're trying to solve. Interesting observation to make here, I think. The field began, you might say, of chemical evolution in 1953 with the publication of Stanley Miller and Harold Urey's paper. Well, really at this point, Stanley Miller's paper on the origin of life, showing how you could simulate in a laboratory glassware conditions that might have existed on the early Earth some 3 to 4 billions of years ago. In that same year that Stanley Miller showed how life might emerge from simple chemistry, Francis Crick and James Watson startled the world with the publication of their structure of the DNA molecule. So the whole genetic revolution, you might say, has begun in how complex we now understand increasingly life is, even at the simple, quote, simple level. It's far more complex than anyone ever dreamed at the time that the first model of chemical evolution as a way to explain how life began was presented. So you have the idea that first chemical evolution is going to show how the first cells began, and is increasingly, since that time, we found out level after level after level of inquiry that the simple cell is far, far from simple. And then it's just the pure complexity of the subject has shown that these crude blasting of chemicals technique with ultraviolet light or heat or any other type of energy source is just conceptually, it seems, increasingly the wrong approach.
Do you see a particular irony in the timing of Stanley Miller's experiments and the discovery of DNA by Watson and Crick?
Well, irony, yes, in the sense that one is trying to show how simple life is. The other one shows how utterly complex life is. And the two methods or approaches just don't seem to fit.
How does the emergence of modern genetics tie in with the Darwinian scenario of life going from simple to complex?
Well, first of all, you have to redefine what you mean by simple. At the time of Darwin, the cell theory, for example, was just a few years old. And so his whole thesis from start to finish did not take into account what we would now call the complex cell. It did not consider that at all. So you have to redefine what you mean by simple, and further analysis has shown that there isn't anything simple. There's no one in the world that can look at a bacterium, for example, and can say that this is simple. We really don't know that it's more simple than even a multicellular creature, and depending on how you define it. The inner workings of the cell are just as complex. Just as many enzymes in a bacterium as there is in a multicellular organism. And that's an unusual discovery, but that seems to be the way things are.
What are the major problems with origin of life simulation experiments?
Well, let's look at the prebiotic simulation experiments just for a minute, because you have to understand what is being done in these experiments. The goal is to simulate in a laboratory setting the conditions, as the theory says, of conditions that might have existed on an Earth 3 to 4 billion years ago. Now, in principle, there's nothing wrong with this approach. However, in order to get experiments that work, you have to constrain the conditions so that you can get results. Even the design of the original Miller experiment illustrates this. He arranged the flask in one way and no results. And you rearrange the glassware and you get results. So it was, why were the results, why do we consider the arrangement that gave results that set of conditions which match the early Earth instead of the other one? Well, it's because the one set of conditions, the glassware, gives results that matches the theory. Now that's fine. Then you have to go on and consider, well, what else are you doing in this simulation experiment that corresponds with what you say existed on an early Earth?
Isn't it rather impressive that amino acids were produced in the Miller experiments?
Well, that's what was so impressive to many off the bat. You take a simple environment of methane, ammonia, water vapor, and hydrogen gas. No oxygen in these systems, by the way, because according to the chemical evolution hypothesis, oxygen would only appear on the scene after the first life began to produce oxygen. The first plant life would have produced oxygen. And then you can do these experiments without oxygen there because you're dealing with it prior to the beginnings of life. At least that's the idea. It turns out there's a practical benefit of doing this. You don't get experiments to work if you have oxygen present because oxygen is a poison preventing these type of chemical reactions. Another thing that is that if you use methane and ammonia, in other words, the so-called reducing environment with some hydrogen gas present, you can get plenty of organic materials produced. And yes, it was very encouraging to the proponents of the theory when amino acids, a few amino acids were discovered in the flask. In fact, over the years, more than a dozen such amino acids have been found in the Miller type experiment.
How close is the development of amino acids to the threshold of life?
Well, you have to have many things besides amino acids. And if you only produce the suite of amino acids that are necessary for living things, and there is a subset of 20 amino acids that we know in living things, you need those, but you need many other compounds and chemicals. But let's just deal with the proteins or the amino acids as their components first, because in these simple experiments, simulation experiments, you produce not only that set of amino acids, but many other amino acids. There are far more amino acids that are produced, and even in greater abundance than are produced of the 20 amino acids. And so you mix all those together with all of the other chemicals that are being produced, and you find out that the interfering cross reactions of these just prevents any type of meaningful buildup of chemicals that are sufficient to go on to the next step in making life. So it's a very difficult, almost intractable problem, I would say, chemically.
What are the steps involved in producing proteins from amino acids?
Well, the way typically these experiments are done, you do an experiment, you produce a glycine, an alanine, and a few other amino acids that we know exist in living systems, in true proteins, for example. And then you isolate those amino acids from the system, and you purify these amino acids, and then you use these amino acids separated, isolated from the myriad of other chemicals that would have been present in a, quote, prebiotic soup of chemicals. And then you react these amino acids together under other conditions to produce polymers. In other words, you're trying to build up, see if you can simulate the production of a protein.
Why are amino acids isolated during this process?
We want to separate the chemicals from each other in different stages of simulation experiments so that we can get a realistic handle on how the developmental process of chemical evolution occurs. If you don't do this, you have all the different chemicals mixed together and they will mutually interact and react with each other. Now, why this is bad, or what happens is, amino acids will combine with amines or other groups that have say a carbonyl group, and you will get dead end process. You won't get meaningful substances.
What do you get?
Just a goo, just other substances that don't have any biological significance to them. And this is the really difficult part in trying to conceive how to do a simulation experiment. It's well known, nearly every one of these experimenters know that you can't do a realistic experiment according to what the theory says and get meaningful results. But if you just did that, everybody would get the same glop, and it's not a meaningful result. And so you have to have meaningful results to publish, and that's the reason why you separate things. So you can get have an idea of how you're going step by step to produce, to go along the process of making life.
How can the investigator affect the outcome of a simulation experiment?
Well, you must have, in any kind of a simulation experiment, the role of the investigator. I mean, it's impossible to do experiments without the investigator. So when we wrote our chapter on the role of the investigator in these simulation experiments, we were trying to point out and make a distinction between those legitimate interferences in the experiment, such as stopping the experiment at a certain time and withdrawing sample for analysis. That's legitimate. You can do this. The experimenter can set up the laboratory conditions and set the parameters of the experiment, and that's fine. These are the kinds of investigator role that you would expect. What is illegitimate, and we felt like needed to be brought to the attention of students and other professors who were investigating this and brought it out into the open, because, believe me, it's never brought out into the open. You can go to these international meetings, and there's two things that are almost never brought into discussion. One is the problem, as we've already mentioned, the role of these interfering cross reactions. If you discuss the role of the interfering cross reactions, there would be no other place for the conference to go. You would just focus right there. It would stop. There wouldn't be anywhere else to go because you can't have any meaningful results. So nobody talks about it. That's frankly the way it is. The other thing is that besides the interfering cross reactions, you must not talk about in these conferences the role of the investigator to interfere in an illegitimate way with results, because if you did, it would become obvious that most of these experiments are unacceptable according to their own criteria for what we're doing is prebiotic simulation. That means, by the way, let me explain what I mean by that. According to the theory, we want to reproduce in a laboratory setting conditions that would have been on an early Earth 3 to 4 billion years ago, and that means no interference from God or any other outside forces, and either then or since. So we make a naturalistic assumption from the beginning to the present. Well, that means then that we cannot have, if we're going to simulate those conditions, we cannot have an investigator constraining the conditions that are outside the boundaries of what we've already said were acceptable for that prebiotic Earth.
How did you evaluate the different chemical evolution experiments?
Basically, we put the scale in such tentatively drawn. I mean, if we can show that it fits the parameters of a legitimate prebiotic experiment, then yes, by all means include it. And we'd be glad to move our line one way or the other according to how the data goes. But so far, the experiments are such that the investigator's role prejudices the outcome of the experiment in an illegitimate way. I just find it intolerable that men of science can allow this to go on and not make it vocal and not make it open, and certainly not, be more specific in their research papers. As I mentioned earlier, though, the reason why we don't do it, why it's not done, is that if you did, you wouldn't get meaningful results. And you can't publish a lack of results. You can't just do a research paper and say, well, I'll publish a paper of what I've done the last 2 months that hasn't worked. You've got to have some results. And it turns out that in 40 years of all these experiments, by and large, now there are a few exceptions, but by and large, most research papers have fit the category that we would call illegitimate, because the experimental parameters are constrained in such a way, the conditions are constrained in such a way that it would not fit a prebiotic Earth. And that's the primary problem that I see with these simulation experiments.
Are the initial conditions in the simulation experiments plausible?
Okay, let's look at the initial conditions of the early Earth. It started out first that the hypothesis was an atmosphere of methane, ammonia, water vapor, and hydrogen. You would have had hydrogen sulfide also, which you need for certain proteins and other substances that contain sulfur. But originally, the famous Miller experiment only contained methane, ammonia, water vapor, and hydrogen. Let's look at why methane and ammonia are in this experiment. It turns out thermodynamically, that is the energetics of the system that you're dealing with inside the flask, carbon that is in the reduced state, nitrogen that is in the reduced state, and that means the maximum amount of hydrogen connected to the atom, carbon or nitrogen. In those states, a reduced state, it is like having a molecule at the top of a hill. And what happens if you blast it with a spark or of electricity or ultraviolet light, or even heat it up to such an agitation point? Well, it's going to tip it over the edge of the hill, and it'll go downhill and bring about a chemical reaction. It's what is known as a chemically spontaneous process. So in this kind of an atmosphere with methane and ammonia in it, you can blast it with these energy sources, make the reaction go downhill, and bring about a production of amino acids. That's all straightforward chemistry. Fits the physics perfectly. Understandable. The really difficult part is trying to do this with carbon monoxide, or even more realistically, according to more recent investigations of what an early Earth would have looked like, carbon dioxide and nitrogen gas. Very, very poor results. If the early Earth was like the modern geochemistry picture of an early Earth, containing carbon dioxide and nitrogen and some water vapor, it would be extremely difficult that this process ever worked. But what happens is, in order to even get things to work with carbon dioxide and nitrogen, put a little hydrogen in there. We need to have hydrogen. Why? Because when you blast it, you can still make the process work and get some products, but you must have the hydrogen present. This is a doubtful procedure too, but it does get around the problem of not having, trying to deal with the problem of no methane or no ammonia on that early Earth, of which there is no geochemical evidence that it would have been there.
What did the Earth's early atmosphere contain?
Well, for example, the picture that geochemists give today is that look at the effluent gases of volcanoes would give you a pretty good handle on what what kind of gases would have been coming out of the early Earth, and it turns out to be carbon dioxide and nitrogen being produced.
Will the simulation experiments work with this atmosphere?
That's what I'm saying, that you get a very, very, very small amount of any organic compounds in this kind of environment. You must have hydrogen there, and it really does help. And in fact, Stanley Miller has said on a number of occasions, well, I don't care what the geochemists say, you can't do the experiments without methane, so since we know that life is here, life must have had, these early Earth must have had methane on it. Now, this is the kind of reasoning that really dominates in certain quarters of origin of life studies. It's not the typical scientific approach that you would expect and you would like to see. It is dominated by a metaphysical condition, I would say. We know naturalistically that life is a result of these processes of chemistry. Well, how do we know this? Well, we're here. And so how can we get here? Well, we've got to have certain chemical reactions that will lead us to where we are. And what kind of conditions? Well, the only way we can make it work is if there was some methane and ammonia there. Therefore, they must have been present. So this must have been argument is used very often in this field, and it's very frustrating for those of us who are trying to move the field over to more realistic scientific approach to this problem.
There seems to be an underlying assumption that the origin of life resulted without any intelligent input whatsoever. Yet the simulation experiments appear to rely upon intelligent guidance. Could you comment on this irony?
Well, there's an interesting contradiction that comes in when you look at the stated assumptions about what the conditions on the early Earth were and what is required to make an experiment work. You have an early Earth, says the theory, void of any intelligent activity. That's by definition to do a naturalistic experiment. But in order to make the experiments work, the illegitimate role of the investigator, who is always intelligent, prejudices the direction or changes the direction over which it would normally go if that investigator did not do certain things. Now, let me give you a specific example here, because I think it's important for it to be understood how it works. The theory says that, for example, ultraviolet light would have been a major energy source on the early Earth. There's no reason to doubt that it wasn't irradiating the early Earth. So we want to simulate in our laboratory setting experiments using ultraviolet light. Now, in order to do the experiment, even with methane and ammonia present, you must use the shorter wavelengths of ultraviolet light, less than 2,000 angstrom units or wavelengths, in order to make the chemical reactions take place to give us a product. But what happens if you use the longer wavelengths, say greater than 3,000 angstrom units? What happens if you use those? If you use those, say greater than 2,000 angstrom units? Well, what happens is that the destructive processes of the products begins in very big time. And so you're destroying the products of this reaction thousands of times faster than you are producing the products. So in a laboratory setting, if you use the full spectrum, for example, of course experimentally you cannot do this. We don't have a way to get the full spectrum, but we can produce longer wavelengths and shorter wavelengths. But these experiments are never done this way. We filter out the longer wavelengths, and I say that's illegitimate.
Are there any natural processes that would have filtered out destructive ultraviolet light?
If, in fact, there was a realistic filtering mechanism, then of course it would be an acceptable experiment. But no one has suggested any way, any possibility or any mechanism for eliminating the longer wavelengths and just allowing the short wavelengths to react with the materials. And I think that, frankly, it's done because it gives the results that we have already specified as a stepwise procedure toward the development of living things. And interesting thing was this, another factor that must be considered, I think. By the time a body of criticism in the literature of origin of life was built up, okay, to show that these destructive processes would have far and away dominated the situation. By the time this literature was built up, the assumption was already set and permeated the whole field. We can do it. The monomer level, in other words, the first level of making amino acids, is presupposed nearly by everybody. And so the fact these criticisms are unheeded. And so we've gone on now to explain, well, how do we make proteins? The first 10 years of the field, say from 1953 until, say, 1965, 66, almost all, the vast majority of papers during that time was, how do we make amino acids? How do we make adenine, one of the bases in DNA and RNA? How do we make the sugars, the monomers, because you can't have a DNA or an RNA if you don't have sugars? Well, despite all of the body of criticism that says, well, it's not very realistic that you ever had a prebiotic soup that had adenine in it or ribose in it, or that had very meaningful concentrations of these various amino acids from which you can make proteins. Despite that criticism, the field today has presupposed that we have granite to stand on. And they don't even have sand to stand on. And they're trying to build polymers, proteins and nucleic acids, as if we have solid evidential basis that the precursor or components on which to build these polymers has already been established. And it has not been established. So the field now is dominated, majority of papers are dealing with the polymer level. So we're in a different decade and we're doing different things, but it hasn't worked out.
What additional steps would be required to develop life?
In order to have life, you have to have not only the amino acids from which you make proteins, and the nucleic acid, the various components to make nucleic acids, but you have to have the the assembly for the cell structure, cell membrane. And the cell membrane is not simply a bunch of fatty acids that react and make a micellar or a bilayer of chemicals, so that you can have an enclosed boundary. What has been discovered in the last decade about the cell wall, the cell membrane, to show that it is utterly complex, extremely complex. It's perhaps one of the major discriminating factors in allowing certain materials in and out of the cell. Now, this structure must be there in order to have a viable living system. And no one has any clue how to do this. There are many things of which we don't know how to put together, how to assemble it. But conceptually, the problem has been for the last 25 years to make the monomers and then to see how to make the polymers, and then how to, and different research workers and groups are trying to work on different facets of the problem. But let's not kid ourselves. We're discovering more and more how utterly complex the living system is. And that's why the field is shifting and changing. We're either at the end of a paradigm where chemical evolutionary processes are being recognized as is the wrong approach. I mean, however much merit there has been seen in the past, we're rapidly approaching the age where more and more research workers are saying, well, we're going to have to try something else. We're going to have to do something else. Why? Because no results that are meaningful. There's not anybody in the world that can explain in a realistic way how to make a protein. There's not anybody in the world that can offer a realistic explanation for how to go step by step to make even a nucleic acid. And in a realistic environment now, with plausible conditions, And I have to put that qualifier in there with plausible conditions, because people get Nobel Prizes for using constrained conditions, where you manipulate the conditions so that the chemistry only goes in one direction. But if you do this, that's no longer prebiotic. You see, that's the problem. We have to do it in such a way we can mimic a dumb, stupid nature, blind, chemical, physical processes. Now, if we are going to require something of the level of a Nobel Prize winning scientist to manipulate the conditions, then you can do this with great difficulty to make some things, but still no one's ever made a bacterium. No one's ever made a cell of any kind. No one's ever made a virus, even. No one's ever made the kind of protein from just basic chemistry. Now, it can be simple chemical steps. Now, Fred Sanger and others have gotten Nobel Prizes for making, like, for example, insulin, but certainly this was not done with the kind of crude processes that the origin of life field is trying to use to mimic the process.
What is so difficult about making proteins or nucleic acids?
Well, the problem in trying to make a protein, for example, from even if you have just a pool of amino acids, is that the energetics is such that now it's an uphill process. Now, I mentioned before having methane and ammonia and blasting them with the energy and having it as a downhill process. Well, let's assume now all we have is a pool of amino acids that are the right set of amino acids to make a true protein. Well, how are we going to assemble the units, individual amino acids in a linear sequence to make a protein? In the first place, in a real protein, they're all left-handed amino acids, I guess you would call it, except for glycine, which doesn't have any handedness. But all of the other 19 amino acids are left-handed amino acids. Well, when you do the simulation experiments, you get a 50-50 mixture. Now, there's no way around that anyone has found. If someone could do a laboratory experiment and say that fit the conditions of something realistic on an early Earth where you just made left-handed amino acids, but this would be a tremendous encouragement to the field. In fact, that person would probably get a Nobel Prize. For example, if somebody can get a Nobel Prize for showing that an enzyme can have catalytic activity, then which is a requirement. What did I say?
RNA.
Yeah, RNA is now recognized to have catalytic activity. It's not just a part of the nucleic acid or part of the informational system, but it is recognized to do the role of, on a somewhat more limited level, of an enzyme. So if this can happen and is worthy of giving a Nobel Prize for this discovery, then certainly anyone who figured out a way to produce left-handed amino acids would probably be worthy of the same kind of thing, especially considering the amount of work that's already gone into it. Now, that's one thing. The other problem is linking them together. In a real system, you can't do it without having an enzyme present. In fact, the assembly process it can't happen without enzymes. And the reason being, you must have the energy put at the right place to make the reaction go uphill. And that's the problem. It's a tremendous uphill. It'd be the equivalent of, I guess, being down in a valley and trying to go to the top of a mountain. And this doesn't happen without not only energy. If you put energy if you have gasoline, you have energy, but you have to have it in the right kind of engine. I mean, you don't just pour gasoline over your some kind of some kind of a wagon. You've got to have an internal combustion engine to use the gasoline to drive you up the hill. That's what the enzyme does. It uses the energy that you have available and couples it to the individual amino acids to bring about the production of the protein.
In addition to the energy problem in protein synthesis, isn't there a sequencing problem?
Absolutely. In order to have viable protein, you must have the right sequence. And the reason for that, according to the standard way of looking at it, is if you don't have the right sequence, the protein doesn't fold up properly to give its overall three-dimensional structure. Best way I can describe it is that the overall three-dimensional structure is said to be held within the sequence of the individual amino acids in the protein. If you had the right sequence, the molecule would fold up. Okay, so I call this self-assembly. But you must get the right sequence. And you can't get the right sequence by just chemical processes that aren't directed. And they are directed in living system by the enzyme. But in a flask, they can be directed by the role of the investigator. So you can't get around the fact that either you have to have an enzyme or you have to have the role of the investigator. If you have the role of the investigator, that's an illegitimate entity for what you said is the prebiotic experiment. And that's the impasse this field has been in for some time.
Are DNA sequences analogous to a written language?
Probably one of the real emotional experiences, you might say, that I've had in doing this kind of work over the last 20 years is I used to have almost every biology book I've picked up and trying to explain how the DNA molecule works in helping to code for proteins, for example. How to make proteins by using amino acids. The explanation has been given in terms of the analogy to a written message. And it is, of course, in terms of function, this is precisely what happens. And after, I guess, a number of years of reading the literature on these origin of life experiments and thinking about how they were worked out, and then focusing in on the role of the investigator and thinking about what the criterion is, or the criteria are for making successful experiments, and realizing how the enzyme works actually in the assembly process, and just figuring all of this together and then being hit with, well, it's only a crude superficial resemblance to a language, I mean, in reality, but it looks like one, but it must have been the result of just purely natural processes here. I began to see that as increasingly unrealistic. That it would be comparable to going to Old Man of the Mountain in New Hampshire and recognizing the face on the side of the hill, and then going to Mount Rushmore and seeing the faces of presidents on the granite and saying, well, these also were the same kind of superficial resemblance. Well, it's obvious. When you see it, you know wind erosional processes are not responsible for putting the faces on Mount Rushmore. To me, it's the same problem we've got when we look at the informational sequences in DNA. Most of my colleagues think this is purely rubbish, and they say this is the same kind of superficial resemblance as Old Man of the Mountain, and I don't think so. It seems to me that what we've got is that the requirements are too close and the similarities are too great for it to be just coincidence. So I'm very much impressed by this, and I'm still open to consider a purely natural process that can explain it, but I haven't seen anything that makes sense so far.
Has Hubert-Yaki made similar claims?
Yes, of course, Yankee's paper did not actually make this connection. It was something he did say, though, that caused me to think more realistically about the idea that the information content is not just a superficial resemblance to a language. Here's what he said, if I could remember it exactly. He said that there is a mathematical identity in the structure of these base sequences in DNA and the alphabetical letter sequences in a message. So he's only talking about a mathematical identity of the structures of these two systems. Now, by experience, we know that it takes intelligence to arrange the alphabetical letter sequences in a written message. So if they're mathematically identical structures, it seems to me quite reasonable that it also takes intelligence to arrange the base sequences to spell out their genetic message. And that's the basis for this. And until I actually saw that written there like that, the mathematical identical structure, I don't think it ever clicked with me to get beyond the, well, this is just like Old Man on the Mountain. I now realize that what we have in DNA is more like Mount Rushmore.
In the mystery of life's origin, you refer to order, randomness, and specified complexity. Could you give us an overview of these concepts?
Yes, it's very important to make the distinction between order and complexity, and then between complexity and specified complexity, so I'll do that. A random situation is something that you can use, for example, with a sequence. Just pull letters out of a hat at random, and then the sequence would be a random sequence. This is what you would get if you do in the simulation experiments and you made a polymer. There might be some crude ordering. Some people are very impressed with the slight ordering tendencies that natural laws would give in sequencing these things. But of course, it's not like order that you have in a crystal. So an order would be like A, B, A, B, A, B, A, B. This would be a sequence of order. And a random sequence would be like A, J, B, Q, T, something like that, where each letter, you might have a run of 3 or 4 that are consecutive A, but in the long run, each one has equal or near equal probability of appearance out of the sequence. Then a specified sequence is one that has like a written message, T, H, E, D, O, G, the dog, and then spell out in the sequence, the dog ran. This is a sentence that's spelled out, fits the vocabulary, fits the grammar, and the proper syntax. It is this kind of distinction that you can recognize as a way for order, and then randomness, and then specified complexity. Now the thing is that in the examples are that the results of stochastic chemistry, what happens in a lab, in a flask, is an example of a sequence that would be ordered. Would be random. Complex is a random state. Order would be like a crystal. Everything is specified in a particular sequence, a defined arrangement. But specified complexity, it's complex. Some have said this. It's like a random situation where it has certain rules that are like language rules. So it's not a totally ordered arrangement like A, B, A, B, A, B, but it fits the characteristics of a language. And we see that in a language system, but the same thing happens with the DNA and protein. It also happens with making, for example, making a computer, or anything, artifacts, or other examples of specified complexity. Mount Rushmore would be an example of specified complexity. In fact, you don't find examples of specified complexity except the structures of artifacts that people make, and DNA, protein as examples.
What do you think the presence of specified complexity in a living system indicates about its origin?
In terms of the origin of specified complexity in living systems, I would say that if we had any examples available that unrestrained or unconstrained natural processes produced specified complexity, then I would be prepared to go along with the community of scientists that are saying that natural processes produce living systems. But we don't have anything like that. Every single example of specified complexity we have is either an artifact produced by a human being, or else it is the result of the living system. And so, like the structures of DNA and protein fit the pattern of what human beings produce. One way you can look at it, it's easy to understand why for so long people have used the analogy that a human being or a living system is like a machine. The machine analogy to living systems has had a long record now, and it's been very productive in a lot of things that have been accomplished in scientific approaches. But the machine never arrived at by spontaneous equilibration of chemistry and physics. These forces must have certain boundary conditions in which they operate. And this is what human beings do to make artifacts. We can make machines ourselves, but we don't find machines arising by natural processes. It's the same situation. So I would say that really we haven't progressed much in terms of our understanding about this question. So I still conclude that the data supports the position that specified complexity is somehow, whether we like it or not, the result of intelligence.
In inferring the necessity of intelligence to produce life, haven't you ventured from the realm of science to religion?
Oh, I know that everybody says this is the adventure of religion, but you simply look at the structure of what you're dealing with. It fits the pattern of experience has taught us only results by intelligence. All it takes to show that this is not the right answer is to find a single example of a natural process that produces specified complexity. And then we would have to really rethink things. For example, the example of William Paley is in the watch is classic. Everyone has heard about this. You find a watch and you know that someone with intelligence has been there and made the watch. Well, this is recognized, of course. Now there's an argument that is often given to explain this in the chemical evolution crowd and literature. And it goes like this. Well, we know that it takes intelligence by way of someone to make the watch. If we found a watch, we would know that. But this is inorganic materials. We're dealing with a living system, which and then the fact that we have to have human beings that manipulate the conditions to make these experiments work and publish them as prebiotic chemistry experiments. Isn't this a violation of the rule of naturalistic processes? Well, no. Why not? Well, because natural selection produced the investigators. It's a circular argument again that I'm trying to address earlier about we know that the early Earth must have had methane on it. Why? Well, we can't get the experiments to work without methane. This is no help. This is not a scientific approach. This is pure philosophy. And so, yes, I would say that my view is not only empirical and fits the data, but it fits our experience, that intelligence is the direction we ought to be looking for accounting for the information in the living system.
Could you summarize the reasons why you believe intelligence was involved in the origin of life?
The overall complexity. It's such an inordinate level of complexity, plus it fits the pattern of my experience. We don't find anything like this, the result of natural means and processes. It's only the circular argument of saying that we are here by the result of natural selection, which produces, in other words, natural selection produces us, we produce artifacts, therefore we don't have to worry about the fact that the watch is produced by an individual, and so we don't have to worry about the successful experiments because we find that human intelligence was produced by natural selection. Well, you go around and around in a circle. The fact is, we have such high-level complexity. Where do we find it? In living systems. Now, we have to have some way to break out of the circle, it seems to me. Now, you either have to rule this answer by religion or some other philosophy, And I say that by and large, what is being happened right now, by the failure to follow experience, we are demanding that our students and our professors only look at natural process, and it doesn't fit our experience, and I think every researcher in the world knows it. It knows that it fits the pattern of Mount Rushmore and a watch. This is not the issue. The issue is it's unthinkable it could be anything else but natural process. Why? Well, it must be natural process. This is philosophy.
How is the mystery of life's origin received by the scientific community?
Well, I think when the book came out, of course, we were a bit apprehensive. In 1984, when the mystery of life's origin hit the world, we might say, there were very few people who were acknowledging the kinds of things we were saying. Well, in 9 years, or then a decade since then, there's been a tremendous shift. The kinds of things that we've talked about in this book are generally regarded more and more favorably among the researchers. They know that the chemistry is not favorable to the production of living systems. It doesn't go that way. But it's standard textbook orthodoxy nonetheless. But in terms of going to the Origin of Life meetings, and interacting with the researchers who do the work, they're very candid. They're very, very upfront, and they know what's going on.
What are the major objections to your current point of view?
They say you can't do research if you hold design. It's really a strange kind of logic that works. We're going to hold a theory that natural processes produce this, and continue to look for natural ways to explain it. And by the way, I want to put a parenthesis in here. I'm not at all suggesting that we should stop research on natural process approaches to the origin of life. I'm not at all suggesting this. In fact, I think it would be a huge mistake if we did stop that. But I am saying that while we pursue natural process approach to the origin of life, it ought to be that the field should be wide enough to entertain other possibilities, other approaches. If these don't work, let's look at some others and maybe continue to work this. Maybe one of these will work. But it's sort of like the idea that if you say that only natural processes are at work, it's sort of like taking your geology students to Mount Rushmore and saying, look at this mountain. Come up with some wind and erosional process or other set of natural factors that would account for these faces. I'm saying that we have to be guided by the structure of the effects we see and let that determine the course of our inquiry, not the other way around.
What was Dean Kenyon's response to your critiques of his book, Biochemical Predestination?
Well, the first contact that I had with Professor Kenyon was a letter after having read his book, and a few years later, having done my own original response to it. And I contacted him with an essay that I had written. It wasn't the manuscript for the book, it was an original essay that I had written prior to that. And he wrote back and was very positive to the criticisms that I had given, and suggested that he too had changed his mind about a lot of this and encouraged me to continue. I think that his shift had already happened before I contacted him, but certainly he was a big encouragement to me in terms of continuing along. You can imagine what it was like. I felt like myself, I was virtually alone out there. There may have been other critics who were saying these things, and obviously Kenyon was, but I hadn't read any of it, and I couldn't find anything in print that supported the views that I was coming to. So I was very tentatively putting forth, sort of like a child going to the ocean, never been to the ocean before. You know, and you're afraid of this. You don't know whether you want to get out there and venture forth or not. And that's frankly the way I felt about the opinions I was coming to about the origin of life field and evaluation of the experiments. So when we presented the essay to Kenyon, and he was favorable to that, I began to correspond with other people with one of the drafts of this book. And we heard from a variety of different people who were doing research in the field, and they gave very valuable feedback. And what I really expected to find was an avalanche of resistance and criticism. And do you know that we did not find it? We found that by and large, the chemists agreed with the chemistry statements, the geologists agreed with the geology statements, and the engineers or the physicists agreed with the physics statements. But here's what I call the Swiss cheese mentality, or the Swiss cheese view of knowledge or science coming into play here. Someone who was an expert in chemistry would see what we had done there and would say, well, yes, that's fine, but your problems are over here in geology or physics or something, or biochemistry. And then someone else who was expert in geology or physics would read those parts and would say, well, those parts are fine, but your chemistry needs to be worked on over here. And what we've discovered was that people's knowledge in their own area was up to date, but their knowledge outside their discipline was behind a few years, and they hadn't realized that enough criticism over the whole field had been such as to basically question the credibility of this whole enterprise as the dominating paradigm for how life occurred on this planet.
What was his response to the mystery of life's origin?
That was an interesting experience. Let me tell about the time that I first met Professor Kenyon and the response that he gave to the book. I had wanted him to write the foreword to the book because his own book had stimulated my own thinking so much in the beginning of it. And I went to California in San Francisco State and met him after he had already had the manuscript for some time. And went in to see him, and he was very no clues given with body language or gestures or smiles or anything like this that he accepted the point of view of the book, and I was really somewhat apprehensive that how he might have found all manner of objections that would just undermine the whole thing, because at that point, as I mentioned, we still felt fairly alone out there and stranded and isolated. And after a few minutes of just quiet in his office and discussion about general things, I said, well, Professor Kenyon, what's your response to the book? And he was just became very animated after a few seconds. He says, I really like it. And I don't know what caused me to say this, and my first response, well, then why don't you write the foreword to the book? And as I can remember, he said, well, I was hoping you would ask me that. And so that's how he became to write the foreword. But he was very favorably impressed with the overall treatment. He had some critical comments to make here and there, and we addressed those. But I think that he felt like it was the kind of response that he wanted to give, but because of administrative duties, other things, didn't have time to go back and make those changes. I hope he will revise and update his own book, because it was certainly a masterfully done work.
Were you a bit apprehensive about meeting Kenyon after writing a book which was critical of his views in biochemical predestination?
Yes, I was apprehensive about meeting Kenyon that first time, because I wasn't sure how he would receive it. I mean, if in fact he had been where he was with biochemical predestination, I would have imagined it would come across overall as a very devastating jolt to his prior position. and it certainly was. But as he said, that over the years, in fact, he said one thing that was very interesting to me. He said even at the time they were writing the book, he was aware of these problems. And I think all of the researchers are aware of these problems. It's not that they don't know. I think the problem is that the dominant perception or philosophy of the culture is such that it's not attractive to question. And you must go on and do research along the old paradigm, even if it doesn't give us the results that we know meets our experience. And fortunately, there's a growing body of people who are beginning to question these things and to come out. But the whole paradigm itself is effectively dead, in my opinion.
Are self-organizational theories plausible?
Well, there are a lot of people who are still doing the self-organizational work, as you mentioned. I think, frankly, though, there aren't any realistic self-organization studies that have been done. When I say that, I mean that they presuppose that you have gotten past some way, you've solved all the problems at the monomer level that I've already discussed. And that's why I mean that there are not any realistic ones. And that you just don't have examples that fit what we say must have been like without an investigator interference.
Would you comment on the work done by Prigogine and Eigen?
Prigogine's work has often been looked at as a solution to the problems we've talked about. But Prigogine's work doesn't solve these problems. And here's why. Prigogine is talking about specificity. He can produce this in his system. He can produce, but he cannot produce specified complexity. And this is the real issue. We're not trying to produce a system that can just get nucleotides to form. You've got to produce them in the right way. And not even Prigogine has solved that problem, even of producing the nucleotides, the polynucleotides. He's done a very good work. I first learned about Prigogine as a graduate student, learning about his irreversible thermodynamic processes years ago. And of course, after he had developed a lot of this area of work, and it was a legitimate bona fide Nobel Prize he received. But people, I think, are unduly capitalizing on that, as he has written about the origin of life. He has not solved this problem, and I think even he would acknowledge this. And certainly Manfred Eigen knows that his hypercycle is way too complex. Even at the outset, he is another example of someone who's a Nobel Prize-winning physicist who came into the area of biology to try to explain the origin of life, and has done is offered one of the best theoretical explanations. If you had this high level of complexity, certain minimum level of complexity of what he calls the hypercycle, but again, that is way too complex. And he himself has acknowledged that it would not come, although he writes as if, well, the prebiotic soup would give us this. If you look at his research papers, he says the prebiotic soup, and then it's almost like hand waving, and then we get to the hypercycle, and then we get these other things. No one realistically addresses the key problems of this. The key problems are how to produce, the key problem is of how to make the legitimate polymers, and how to make the specificity problem, how to solve this.
What is your assessment of RNA scenarios?
Some years ago, the suggestion was made that we have an RNA world. Now, the reason why this became a subject of approach was the recognition that RNA itself has some has some catalytic activity. And prior to this, we were trying to look for some way to break out of the chicken and egg, hen and egg problem. You have the fact that DNA and proteins are both required for the living system. The DNAs produce the proteins in a living system, but really, which came first? And in this field of the origin of life, there's a huge faction that says, well, proteins came first. These would be the enzymes. And then there are other crowds said, well, DNA came first. When it was discovered that RNA, as one of the nucleic acids, itself could serve as the capacity of catalytic activity, which the enzymes normally provide, then it said, oh, we've solved this problem. RNAs, or the RNA world, can account for the living system. It was, for a while there, I think it was looked at as perhaps realistic, and there's still some people who still write as if this has solved the problem. But conceptually, or not conceptually, but realistically, on a primitive Earth is where you have to deal with the problem, because you cannot have RNA without ribose, a sugar, and you cannot have RNA without the nucleic acid bases, and adenine and the other bases. You cannot have RNA without phosphates. Well, how do you get that from the prebiotic soup? And this is the problem. In fact, in one of our chapters in the book, The Myth of the Prebiotic Soup deals with precisely this problem. You just have to come to the conclusion, if you know enough chemistry, that, or you take the chemistry realistically. You might know enough chemistry, but you have to overlook it to conclude that you can get RNA from a viable prebiotic soup. I don't believe anybody in the world who realistically looks at this kind of chemistry could conclude that this is the way RNA came about.
What other problems do you see with an RNA world?
Well, other problems with the RNA world is that you must have a whole, not just RNA, but you must have the other associated substances that go along with it. What you would really like, if you were an advocate of this perspective, is to have natural selection operate at this molecular level. But of course, the problem is, and they're forced to this, I believe. These researchers are hedging on the definition of natural selection. Historically, natural selection requires the living system. Well, how do you apply, then, natural selection before the living system? Well, it would help greatly if you could. But no one has really satisfactorily explained how to do this. So what happens, it just gets posited. Well, we had something like natural selection that would have worked prior to the development of living system. Now, what they mean is applied at the level of RNA, for example, and then maybe it would start working.
What does nature select at that purely chemical level? What sort of things are selected? Complexity, order?
Well, that's still, as far as I'm concerned, no one's ever demonstrated that nature selects anything at this level. It's just overall destructive processes in a realistic environment.